my pleasure to introduce Dr. Lucina Uden, who is currently an associate professor at the University of Miami, and she directs the Brain Connectivity and Cognition Laboratory, as well as the Division of Cognitive and Behavioral Neuroscience. And she has been very active in brain imaging, studying a lot of functional imaging. She's been doing a lot of work with linking behavior to brain, and so I think that's a really interesting topic and kind of a hot topic these days as to how well we can do that and what we can see. And she's also been very active in just promoting diversity on the Human Brain Mapping Council, which I think, have you rotated off as well this year? Or are you still on?
I think I'm not on, but I'm still on the diversity committee, but not on council.
Okay, yeah. So we were on for a while, and I just rotated off myself as well. But she's very approachable and has been great to work with, and I think you'll really benefit from her talk today. So without further ado, Lucina, I'll let you take it away. Thanks for joining.
Great, thanks so much, Vince and Armin and everyone for having me here today. As you've heard, I'm Lucina Uden. I'm here to share with you some of the work that our lab's been doing over these past few years, focusing mainly on how brain dynamics might relate to flexible behaviors. And before we get started, I just wanted to acknowledge, these are some strange times, and we're. Oh, can you hear me? Can you hear me?
I can now. Yes.
Hello?
Yeah, I can hear you.
Yes,
you can hear?
I can hear. Mm-hmm. Yes.
Vince, can you hear? Oh, okay, great. Okay, yeah, let me jump in if something goes wrong, because I won't know otherwise. So please feel free to interrupt if the audio goes out. But yeah, I wanted to thank you all for inviting me here. It's really great to be able to chat, if not in person, at least in some way. And these are strange times, so I'm not going to pretend like these are normal talks and where everything's fine. Everything is not fine. And for many of us, they're just it's very hard to focus on work these days with everything that's going on. And one thing in particular, I think it's hard for me to concentrate on such things like publishing the next paper when there's so much social unrest and there's so much to think about and so much to act upon that it almost becomes overwhelming and can be lead to a sense of despair in some situations. So some of the things I try to do in day-to-day life is think about what's going on and think about what power I have as an individual. And one of the things I like is this quote from Toni Morrison that reminds us, if you have some power, then your job is to empower somebody else. And especially thinking in the vein of early career researchers who are just starting their labs, or postdocs, grad students thinking about what they're going to do with their careers. Remember that you're going to soon be those people in positions of power, whether it's the editorial boards or the editor-in-chief of a journal, whether it's the chair of OHBM or the head of your department or your division. Eventually, you'll be the one who is the gatekeeper in your community. So what can you do when you have that power in your hands in terms of empowering others who have been marginalized or who have not had the same opportunities? I think that's the only way for me personally to stay sane in a world where it sounds like what from one day to the next everything is unraveling out of control. But I think we can all do something with the power and the platform that we have. So that's something I like to keep in mind. So what I'll tell you about today is mostly about cognitive flexibility. It's something we've been very interested in for a while, and that's mainly because I can tell you, of course, that these abilities relate to better academic performance and life outcomes and all those other things. They're affected in children with autism spectrum disorder in ways that make transitions during day-to-day life very difficult. But right now we're all experiencing the ultimate in being flexible. We're switching from in-person teaching to online teaching. Some people are doing working from home and figuring out how to get all their things done in a very different situation from what we're used to. That's flexibility in our daily lives. So I don't have to tell you that flexibility is important, and it's something that's interesting to understand in terms of individual differences and the brain systems that support flexibility more broadly. So in our lab for a while now, we've been interested in these big picture questions about how brain networks develop from childhood through adolescence into adulthood, and how that relates to cognitive development, in particular executive functions and flexible behaviors, but cognitive development broadly. And we're interested very much in the consequences of atypical development. So what happens when there is a differential wiring within circuits that are important for development of these flexible behaviors. So the way we do this is by studying kind of neurotypical or typical individuals, as well as typically developing children and clinical populations using a combination of structural MRI, functional MRI, diffusion MRI, causal modeling, and various kind of statistical tricks. And we try to I'll give you kind of a little taste of some of these different studies that we have ongoing. And the whole framework for everything we do falls under this kind of network neuroscience umbrella, which a term which I think some of you are very familiar with. And I really like a paper back from 1990 by Marcel Nesselum that talks about how all cognition and behavior is the result of interactions between large-scale neural networks. So that behavior is really mapped at the level of multifocal neural systems rather than anatomical sites. And the reason I think this paper is so interesting is because it was written in 1990. This was really before all the kinds of things we see nowadays with structural and functional MRI and human connectomics. This was even back in the days of neurology. This was an insight that he saw from examining patients with lesions. So I think that this holds today, that we really need to understand how brain networks are organized in order to really understand behavior. And I also like the work of Randy McIntosh, where he talks a lot about how the functional relevance of a brain area depends on the status of other connected areas or the context in which that network is manifested. And there's also a good review from Luis Pessoa about brain dynamics and brain networks that talks about how a network needs to be understood in terms of interactions between multiple brain regions as they unfold temporally. So all of these are kind of in the background of the work that we do, and that kind of motivates a lot of the questions and the approaches that we take. And of course, as you all know, back in 1995 or so, when Bharat Biswal introduced resting state fMRI into the arsenal of tools that we can use in neuroscience, it kind of changed the game for everyone. So you can see these kind of low frequency fluctuations across systems. We know to be involved in attention, memory, language. All of these are sort of occurring spontaneously in the background, and we can actually study their integrity and configuration and development and how they're affected in clinical populations. So this has really been a game changer. In terms of on the sort of cognitive flexibility end, there's been many years of research coming up with tasks that are very complicated, and putting people in the scanner and asking them to do these kinds of things, which really make them choose flexibly or switch tasks or shift sets from one sort of behaviors to another. Sometimes they're very difficult to translate into clinical and developmental studies because they're just very complicated tasks. So I'll talk about that a little bit. But if you think about what cognitive flexibility is, it really draws upon these cognitive processes like salience detection and attention, inhibition, switching, and working memory. And there's been, many years of cognitive neuroscience looking at the neural substrates of these different processes. There's just a summary here on the right and a couple reviews from our group if you're interested in sort of looking at that further. What we did in the lab was try to come up with something that kids could do, or that children with autism could do, because a lot of the tasks that have been used are quite difficult for those younger than 12 to really understand. So this was one that's adapted from the developmental psych literature, where you're basically asking the question, pick two things that go together in one way. And they could be going together on different attributes: color, shape, size, and then you're going to ask the individual to pick two other things that go together in another way. So then they have to sort of pick a different dimension and flexibly switch their categorization. So in the scanner, if you do this task, we had to come up with a control condition. So here we just say, okay, press the buttons that are outlined. So these two go together because they're both small objects. These two go together because they're both blue, and these two are both representing one object. So this is a task you can titrate in various ways to make it easier or harder. So we adapted this to the scanner environment. And if you look at, this is what it sort of looks like. If you get adults to do this task in the scanner, first we had to do a check to see that the version we did in the scanner was similar to what you would do outside of the scanner just as a behavioral task. So we found high accuracy, high correspondence between accuracy in both conditions. And then we just looked at efficiency, which is a combined reaction time accuracy measure, looking at how people get more efficient at doing this over runs of a task. Now, as one might expect, when you're doing this task and you're comparing the flexible versus the control conditions, you see these widespread frontolateral frontoparietal activations, dorsolateral prefrontal cortex, a lot of basal ganglia as well, anterior insula, and cingulate cortices and cerebellum. So these are what one might expect in a difficult, executive type of task across in terms of brain activation. We were further interested in sort of the task modulated connectivity of brain regions involved here. So in these adults, what we did was this group iterative multiple model estimation, the GIMI method that Katie Gates at UNC has proposed that uses structural equation modeling framework to look at interactivity amongst a user-defined set of regions of interest. And essentially here at the group level, we find that during these flexibility trials, the inferior frontal junction there in red is the kind of the driver of the other brain regions. So even though all those regions are activated by the task, it seems to be the IFJ that is sort of comes online and pushes or drives the activation in DLPFC and parietal lobes and all these other regions. And another finding in this study is great individual variability in the paths or in these connections in terms of this task performance. So in the paper that's discussed in great detail, but I'll leave it at that because I want to have time to get to some of these other clinical findings. So basically, when we then kind of use the work we do in typical development to frame our thinking for how these systems might develop to get to the way we see them in the adult brain. And a whole other line of work in our lab looks at the development of flexibility, both in terms of cognitive and behavioral flexibility and the processes related to those, the cognitive processes related to those, as well as the network interactions and network flexibility metrics that change over the lifespan. We do that by comparing children and adults in things like Granger causal analyses, looking at how they structure changes in certain fiber tracts from childhood to adulthood. We look at how large-scale networks change their topography across the lifespan from age 5 to 85. Look at how these networks, interactions between networks change across the lifespan, and some networks, increase their connectivity with others, et cetera. And we also look at how variability in brain systems changes between the ages of 6 and 85 in ways that we think support flexible behaviors. So this is sort of a separate line of work, but it sets the stage for the studies that I'll focus on today that are more focused on autism. Here, if you look at the children doing the flexible item selection task, they do activate the similar frontoparietal and cingulate insular regions that we see in adults. And this is just a preliminary analysis of activation here. Here's a cautionary tale. This is a registered report that we put in when we started the study, which means, of course, we register the analyses and hypotheses. And then now we're at the position of having to write up the results and publish the full report. And this is 2 or 3 years later. And of course, all the methods have changed, and everything we've said we would do in the original paper is a whole new analytic plan that's more up to date. So that's one of the, I would say, caveats of registered reports, is that whatever you say 2 years ago, as you know, in neuroimaging, is unlikely to be exactly the same pipeline you're going to use a few years later. The other caveat is that COVID happened, and in March we stopped scanning, as did everyone else. So we didn't actually collect all the data we said we would collect for this registered report. So, of course, that is something we're all dealing with right now. All this to say is that the registered report we eventually publish will look very different than what we had initially hoped for, and such is life. So Cognitive flexibility involves coordination among brain regions that we know to support inhibition, working memory, attention, and salience detection. And so we think that adults and children as a whole kind of engage lateral frontoparietal and salience or mid-insular networks during flexible item selection tasks that we've adapted for the scanner. And we see in the adults that task-modulated connectivity of the inferior frontal junction occurs, at least in this task, in the service of flexible behaviors. And stay tuned for that registered report from children with autism, which we'll be curious to see how that turns out.
I think that's an example of cognitive flexibility right there.
Exactly. You will. All of you have had to be very.
Thanks for demonstrating it for us, Lucina.
It's they say you shouldn't everybody just pivot to doing COVID research because we're in the middle of this pandemic, but you've also seen a lot of labs really pivoting their research focus. And I'll talk about one project we're doing at the end that looks at the psychosocial impacts of quarantine on children with autism. So there is some opportunities to be had. But yeah, and thanks for jumping in because I don't know if you can all hear me, so I'm glad if someone just pipes up once in a while and says I can still hear or or something like that. So great. So and this is the part where we get into some work that we've done with Vince's group over the years, looking at the brain dynamics that we think relate to some of these behaviors. And as you all know full well, because it's your toolbox here that we've been using a lot, the GIFT toolbox. You can look at sliding window approaches for functional connectivity, which allow you to, pick, let's say, 30, 40, or 60 seconds of a window and see how the connectivity of the whole brain changes across time, using this approach followed by k-means clustering to group together the different brain states into, things you can sort of give them numbers, basically. And this allows us to kind of look at different metrics of dynamics, such as how frequently does a particular whole brain functional connectivity configuration occur, how long does each particular brain state stick around for the dwell time, and then transitions between states. So how many kind of switches are there between each individual states that we've derived from the clustering. So you all know this. And what Jason Omi in the lab has done basically has been to use independent component analysis to break down, actually this was already done in the HCP data set, the Human Connectome Project, the derivation of these brain regions through ICA gives you a lot of nodes that you can then use for the functional connectivity analyses. Here this was a few years ago, we took the 200 subjects, 1 hour of resting state fMRI data. So you can imagine we could do replication and all that within this same data set because there was so much data to play with. And here there was also a great deal of out of the scanner data in terms of neuropsych, in terms of behavioral testing. And we're particularly interested in this card sort, which is it's a Wisconsin card sort adaptation, a measure of cognitive flexibility. And what Jason found here is that, the brain sort of cycles through various states. I've ordered them by most frequently occurring on the left, 36% of the time, and then least frequently occurring on the right over here, 9.9% of the time. And what you can see is there's this really, there's more frequent occurrences of this attenuated connectivity state on the left, and very few occurrences of these strong connectivity states over on the right. And it turns out that those individuals who spend or who show more frequent occurrences of state 1 and less of state 5, and similarly more frequent occurrences of state 2 and less occurrences of 3, are the ones who do better on the measures of working memory and cognitive flexibility that were taken outside of the scanner. So there's something about these sort of brain states that might, help in these particular types of tasks that require flexible thinking. And, so in this, initial study, we saw that greater cognitive flexibility was associated with the propensity to occupy more frequently occurring states, characterized by attenuated correlations and greater functional connectivity variability. And these are also the states that I think your group has found as related with arousal and vigilance. So there's something about that kind of loose connectivity state that may be related to just greater attentional arousal. And these results kind of highlight the potential for how dynamic functional connectivity approaches are beginning to reveal relationships between brain dynamics and flexible cognition. And then, we kind of wanted to hone in a little bit. So that was a whole brain analysis, kind of agnostically looking at all connections in the brain. Whereas we do have some hypotheses about specific brain networks and how we think they enable flexible behaviors, in terms of their action on other large scale networks. So for a while, we've been finding that, for example, the insula, signals in the insula can be used to predict what happens in central executive and default mode networks. If you use things like Granger causal analysis or dynamic causal modeling, you can sort of see that there's a primacy of the anterior insula in particular. So we think it's actually possibly involved in switching between the sort of actions of these lateral, central executive networks and the sort of medial default mode networks. So we've been kind of looking around at this for a while, and so have other groups. So if you think about this, then the salience network is just, or what I'm now calling the mid-cingulate insular network, is now, been implicated across so many disorders and has been kind of shown to be a driver of other systems in kind of a lot of studies at this point. So we were interested in looking at these particular dynamics in a study that a colleague of mine in Berlin was running. She got these data from about 100 college students, and I'll just show you the test because it's more fun this way. If you're basically supposed to say which letter pops up where the arrow is pointing, and it's going to be an E or an F. But there will be, and there's eye tracking going on during this task. So obviously, if you're cued to look to the left, your eyes will go to the left. However, unbeknownst to the subject, there will be a erotic distractor, and it's actually like much worse than what I'm showing you here. But there's distractors which kind of get your attention. And of course, the eye tracking shows that people pay attention to that distractor, often at the expense of doing the task. And so they might miss that F when it does come up. So you can.
Sorry, Lucina, I missed that. Can you repeat that? I was distracted for a second.
Yeah. Exactly. The erotic distractor works. And so what happens is that you can quantify the eye tracking behavior. Even if people don't tell you they're distracted, their eyes will give them away. And what's nice about that is you don't even need a report other than what the eyes are doing. And it turns out, well, you can get reaction time and error rates and the gaze distance from the target and all these various eye tracking indices, which tell you, yes, people are very much distracted by that kind of thing. And it takes them longer to do the task when such a distractor is on the screen. But what's interesting is that these are behavioral tasks done outside of the scanner with eye tracking, but they relate in interesting ways to the dynamics of these systems in the brain. In particular, if you look at insula and ACC systems, here we just focused on those and looked at their dynamics with respect to the rest of the brain. There's one state here you see, we've called it salient state 4, where that insula and ACC are actually negatively correlated with the visual cortices. And it's that state was the only one where we found a relationship with the behavior. In fact, individuals who spend or show more frequent occurrences of salient state 4 are the ones who are better at the task. So if your visual cortex is sort of in a decoupled state to the salience network, you're actually better at ignoring the distractor. That was something we thought was kind of interesting. And it kind of suggests to us that these dynamics of that system, and also doing the same analyses on the frontoparietal control system, we did not see a correlation there. So something about this sort of bottom-up salient visual processing seemed to be very important, or related at least, to the capacity to resist tempting visual distractors. And this just highlights how specific network dynamic patterns might be related to things like individual differences in self-control. So, if we really get into this is sort of my deep dive into the insula. And the reason this is such is you can study anything you want and you'll probably find insula activation related to it. So emotion, empathy, and pain, affective neuroscientists are always talking about the insula and anterior cingulate. Subjective awareness, interoception, somatosensory processing clearly involves the insula. And at the same time, conflict switching, attention, and inhibition, these high-level control processes, almost always you'll see insula activation in these types of tasks. So you may conclude that the insula does everything, and that you see it for basically any kind of task, or you might try to think about what is the sort of overarching process that it's really engaging that makes it so ubiquitously active. One thing that we've also been interested in is subdivisions and parcellation. Obviously, if the brain region is doing that many things, it's one possibility is that you haven't divided it up carefully enough to understand what each distinct contribution of each region is. So there's been quite a lot of work now, something I'm highlighting here, work from Ben Dean and Luke Chang that suggests you just use resting state fMRI and ask the question, which voxels in the insula have connectivity patterns with the rest of the brain that are more similar to each other than to other parts of the insula. Here they've found at least 3 subdivisions, a dorsal, anterior, a ventral, and a posterior. But I have to say that in these studies of insular parcellation, you can find anywhere from 2 to, I believe, 27 parcels. As you all know, you can get as many parcels as you look for. So, so this is just a caveat on parcellation. But if you do look at these distinct functional subdivisions or treat them as distinct functional subdivisions, co-activation meta-analyses suggest that the ventral anterior tends to be more active in affective or emotional studies and tends to show activation patterns with other limbic regions. So on the left is a study from Luke Chang, on the right is one of ours with Luis Pessoa. And basically on the right is just when the ventral anterior insula is active, all the regions in red are also active across many studies, so they're co-active. And if you look at the next level, when the dorsal anterior insula is active, the studies that are involved in error processing, inhibition, switching, et cetera, are implicated. And also you see lateral frontoparietal systems tend to be co-active with the dorsal anterior insula. And if you get down to the posterior insula, it seems to be more active in the pain, somatosensory studies, and it seems to be co-active in with sensory motor cortices, as you can see on the right. So at the same time, the whole set of these regions I've been talking about is indeed active across all of these different cognitive domains. So there's this common functional fingerprint, even despite the fact that we see these individual subdivisions. So we thought something about dynamics might explain this, like, you know, the fact that the insula is everywhere at all times and everything to everyone. So if you look I'm just going to highlight one finding from this study where we parcellated the insula and looked at the dynamics of these regions as they relate to other parts of the brain. The main thing I want to highlight here is that these different insula states, what happened? Okay, the different insula states reveal that there are some times when all 3 or all 4 in this case subdivisions show similar patterns of connectivity with other regions in the brain. And there's other states where they diverge, and that's the state 2 there. You can see in a small percentage of the time the states diverge, and other percentages of the time the states are more similar. But the thing that I thought was most interesting from all this is that if you compare the dorsal anterior insula, that's the red blob here, with the other insular subdivisions, the dorsal anterior insula is more differentially connected with the rest of the brain. So that's the subdivision where in different connectivity states, you see more diversity in terms of its who it talks to. The sort of connectivity partners are more diverse for the dorsal anterior insula, just consistent with its sort of role as a flexible hub, mediating switches between other systems. And then if you look at the structural connectivity of the dorsal anterior insula using diffusion weighted imaging, you'll find that it has some connections to frontal and thalamic regions that we don't see for the other subdivisions. So at the end of the day, we think there's at least 3, probably more subdivisions, dorsal anterior, ventral anterior, and posterior within the insula, with the dorsal sort of more active during the cognitive control types of processes, ventral more in emotion, and the posterior more in sort of bodily sensations. And we think the dorsal anterior insula is sort of unique and special in that it has more variable functional connections and unique structural connections that maybe permit functional flexibility. The dorsal anterior subdivision is also the one that we found in our earlier studies that sort of causally drives the other systems. In fact, if you use a region of interest in the ventral anterior insula, you don't see that same causal effect. What's one sort of bad thing, as we all know about fMRI, is that it's slow and you can't get this sort of millisecond resolution. You would need to test causal theories, really. And that's the biggest critique of a lot of the modeling that folks do or try to do on these slow signals. But if you happen to be able to get electrodes on the brain, as you can in the case of individuals being monitored for epilepsy, you can sometimes get these, the great temporal and spatial resolution that comes with that. So a lot of groups are starting on that already. Here you see some surface electrodes that have enabled sort of teasing apart relationships between default, attention, salience networks, as I've been saying. What I was very excited about before the pandemic began was this collaboration we've got ongoing with University of Western Ontario, supported by the Canadian Institute for Advanced Research. And this project, this group, these neurosurgeons up in Canada, are one of the few groups that routinely places multiple electrodes in the insula, depth electrodes, in fact, where you can really see that sometimes they really cleanly capture the anterior, mid, and posterior insula through 3 separate leads. And if you can imagine my excitement at seeing this, I thought, well, we can finally really test these subdivisional hypotheses and these network interaction ideas. And of course, no sooner did we get this all up and running than everything shut down in March. So another great project for the future, hopefully. So I think hopefully I've set you up a little bit to give you the final piece of what our lab is very interested in, which is thinking about clinical populations and how the studies that we do in brain dynamics and typical development can help us understand those individuals who are showing flexibility deficits better. And in autism, there's some stats that are really remarkable that every time I see them, I'm surprised. So, about 80% of individuals, children with autism who grew up to be adults with autism, don't actually live independently. They sort of haven't really moved out of their parents' house. There's also this kind of similar grim statistic about an 80% unemployment rate among young adults with autism. This is surprising given that some of these other early life developmental disabilities, you see that they fare much better than those with autism spectrum disorder. I think personally, part of this has to do, obviously, there's social communication deficits in autism, and that's what the main focus has always been in the field. But there's executive function and flexibility deficits in autism, which I think are often overlooked or not as well studied. And if you can imagine in the real world, if you're waiting for the subway and it never comes, you have to take the bus. That's flexibility. That's what you have to do to get to work and to move on with the day. If you're not able to do that in a way that is adaptive, then that can seriously affect your ability to live independently and work full time. So I think the flexibility deficits really deserve a closer look. And there are a few sort of behavioral treatments that can be done in the classroom and with parents that really target those types of flexible behavioral things that you can train up. And we don't really know how they work or why they work in some kids and not in others. So, one thing we really try to do is understand why something might be the case in some individuals with the disorder and not others. And heterogeneity is the sort of story when it comes to many clinical conditions. In autism in particular, here we did an analysis, a latent profile analysis, on a mixed group of children. Some of them were typically developing, some had ADHD, some had autism, and some had autism plus ADHD. And you might think, well, the kids who have the disorders will have executive function deficits. And it turns out, that, as a group, when you do group averages, that may be true. But there's a profile that we see here where some kids are above average on their indicators of executive function. These are all parent report indicators. Some are in the average group and some are impaired. But importantly, not all kids with autism are impaired in executive function. In fact, you see 18% of the middle pie here is kids with autism who have perfectly average executive function and impaired or the comorbid ASD, ADHD group. If you look at, this is a replication study by one of the undergrads in our lab who looked at just our own data of typically developing kids and kids with autism and found a kind of similar breakdown where like a good chunk of the kids with autism still fall into this average executive function group. So you don't have uniform deficits really probably in any domain in any of these disorders. But what can we say about the dynamics that might underlie these individual differences? Is there something unique about the kids that are affected or more affected? Do they show fewer transitions between brain states, for example, as one might predict as a neural correlate of reduced flexible behaviors? And of course, when you do this study here, Jason Nomi in the lab ran this on some autism brain imaging data exchange, resting state fMRI data. So again, the independent component analysis to derive some brain regions. And here we've got 4 states to look at. He did find some initial evidence for reduced number of transitions between brain states. And since this, I don't think we ever got around to publishing this, but since this, there were a number of groups who've shown similar findings. So I think that reduced transitions might be really one of the things that characterize the autism group. Another thing we're doing is looking at coactivation pattern analysis as just a different way of assessing brain dynamics. So here you've got sort of time point by time point grouping of the TRs into coactivation patterns with similar sort of spatial maps. And again, you can still get things like dwell time and transition between states and frequency of occurrence of states. But here, you're not doing the sliding window approach. It's got, sort of different assumptions. But if you use sort of coactivation pattern analysis, you can start to see some interesting things going on in children with autism. So here, Emily Marshall, another great undergrad in our lab, just published this paper where she looked at the salience, or here the insula ACC and singular insular network with the rest of the brain using this approach, and found that there's different brain states where these the salience is sort of coactive with other brain regions, and the state 2 was one in which they sort of central or lateral frontoparietal and salience were sort of coactive together, and that state showed a reduced frequency of occurrence in kids with autism compared to typically developing kids. I'm going to fly through this just because I want to make sure we got time to talk about things, but this paper just came out, so it's available. We're also looking at these dynamics in these salience mid-singular networks as they relate to task performance. So we had a very simple cognitive flexibility task that kids were doing in the scanner, where they're just saying like, which object is different, and it could be different on color or shape. In fact, it was too easy. The task was really easy. But if you look at the coactivation amongst these sort of mid-singular insular, lateral frontoparietal, and medial frontoparietal networks, there's some states or coactivation patterns where pairs of these networks are coactive, and others where they're active on their own. And what we basically found in this study was that towards the end of the task run, so if you have a child doing 4 runs in the scanner, towards the end, of course, they get fatigued. And in kids with autism, it's towards the end of the runs where we see that they have to sort of compensate to keep up the same level of performance. And that compensation sort of comes in the form of the lateral frontoparietal having to be more active towards the end of a task. And that's kind of, we think, making up for the fact that they're sort of losing steam towards the end. So overall, the dynamic functional connectivity approaches here that I've talked about, the sliding window and the coactivation pattern, reveal atypical patterns of brain dynamics, particularly across these mid-singular insular and lateral frontoparietal networks in prevalent neurodevelopmental disorders that we know to be characterized by cognitive inflexibility. And the extent to which these brain dynamics underlie individual differences in flexible behaviors is something that we're currently very much interested in and working on. So all of this is to try to get us a little bit towards precision medicine. There's a review with Manish Sagar I have here last year that talks about all these sort of different ways of carving up data and how we would need to get sort of more deep phenotyping in order to get towards precision medicine. As all of you know who do anything with machine learning, we're pretty far from these goals. But what I like to point to is this Tom Insel paper with Bruce Cuthbert in 2015, where they really have been pushing the RDoC, or the research domain criteria, for some time now, where in sort of a little bit of a critique of the DSM, or Diagnostic and Statistical Manual of Mental Disorders, which could include very mixed groups of individuals that are just based on symptoms. Their idea is that if you take a look at things like genetic risk, brain activity, physiological markers, behavioral processes, and look at the spectrum, across the population and sort of group individuals in a more data-driven way, you might get towards this precision medicine goal a little bit better than the current approach. The only reason I show this figure, though, is because they point to the insular cortex in that figure. And I don't know whether it's because they read all my papers and thought she's, as they just threw a brain region in that figure, but I like to think it's the former. But in any case, that's just my wishful thinking. But this is sort of something we're doing right now, which is totally different than everything I've talked to you about before. Is that, and this is what comes from serendipity again, a student in my lab found that when we were looking at our children with autism, being in South Florida, many of the families are bilingual. And in fact, across the country, Spanish is the second most spoken language in pretty much every state. So we started to think about this a little bit about like, what is there anything about being bilingual that creates a different sort of early life environment for kids with autism that in some cases might turn out to be beneficial. And there's this hypothesis that's been lingering around in the language field for a while that if you're bilingual, you actually might have an advantage in executive functions because bilingualism requires you to switch between different languages, inhibit one language, and things like that. So, of course, there's not been a lot of good empirical support for that in recent large studies. However, there is some support that individuals from lower socioeconomic status do sort of show a bilingual advantage. And later in life, if you have dementia, when you look at the aging literature, those who are bilingual seem to be getting dementia at a later age. So some people suggest that bilingualism buffers against executive function deficits. So there's some kind of interesting thing to study here that we're currently undertaking. A lot of this bilingualism and executive function literature is really pretty new, but some studies suggest that bilingualism can mitigate set shifting difficulties or flexibility deficits. And this is starting to become a field that is really growing. And then, parents are reporting fewer executive function deficits in dual language speakers. The reason this is really controversial and clinically important is that in the language and communication literature, clinicians often say, don't try to teach kids two languages in the home if they have language difficulties because it'll make them more confused. So for a long time, the clinical recommendation was just start with one language because if your child is having language delay, don't confuse them with two. However, that sort of finding and that sort of clinical thinking is sort of outdated at this point, and there hasn't been really any evidence that learning two languages is detrimental to language learning, and in fact might even be beneficial in the executive function domain. And we just had data from our own study because we do this in Florida, and whether we weren't even thinking about it, but most of our participants were bilingual, or many of them were. So we were actually able to break down the kids with autism and the kids who are typically developing, looking at their behavioral rating inventory of executive function, the parent report of executive function. And it turns out, as the literature suggests, the typically developing kids don't show a difference in executive function. The kids with autism, however, we do start to see this bilingual advantage where those who are bilingual have, better, here lower is better. Those who are bilingual in autism had better executive function scores than those who are monolingual. So this is something we're really actively exploring right now, very interested in following up with the brain data. So, what I've started out, trying to tell you is that we're interested in flexible behaviors because, in day-to-day life, if a child only wants to wear one pair of socks and that pair of socks is in the wash, you're going to have a hard time getting the kid out the door if they can't be flexible on their feet. So the ultimate goal of all of the studies I've told you about is to try to find ways of understanding how brain dynamics underlie these individual differences in typical and atypical populations, and how we can get kids to think less about what's on their feet and more about, going out in the world and interacting in the world. So, that's where we want this all to go clinically. And this is a book, if anyone's interested more about the Salience Network and what I think about it. And this is my collaboration network, which many of you are already a part of, and I like that. I love doing that, and I hope that continues to be the way that people make progress in science these days. This is the lab, of course, that does all the work and deserves all the credit. Of course, all the collaborators and the funding organizations that make this possible. It's really a huge thanks to everyone involved. And I think I'm just going to say, just go out and vote, because that's the last or best place that you can actually affect the world today and use your power if you're an American citizen. So don't forget to do that. And this is San Diego, where I am right now. Thanks very much. Happy to take questions.
Thanks, Lucina. Let's see. How can we do questions here? If people have questions, you can unmute and speak, right?
Should I stop sharing the screen? Is that easier?
We can have a screen being shared, so people can ask their question in the chat panel.
Okay.
So I have so many questions. I don't want to be so selfish, so I will keep most of my question for the Slack channel. I just have a quick question regarding the result they show that we have like a different sub region of the insula are involved in different tasks. So you had a slide that shows from different group. Is it from meta-analysis or they are like a task specific? Now I think you are mute. Sorry. I think that we need to unmute.
Hello.
Okay, now I can hear you.
Okay, yeah. I wasn't clear on that slide. That the study from Luke Chang is a meta-analysis, and the study that I was talking about is also a co-activation meta-analysis. So yeah, these are across many studies that are available. Yeah, forgot to mention that.
Thank you. I like this big option. So it seems like that Thomas wants to ask a question.
Sure.
It isn't so much a question per se, but I just wanted to say thanks for the terminology that you use when you're studying individuals on the spectrum. I know some people are back and forth on typically developing, but as a person on the autism spectrum who's known for the last 22 years that they're on the spectrum, it's nice to hear things that just aren't straight up pejorative or moderately dehumanizing, like say we're going to compare to healthy controls, because every single scientist I know on the spectrum genuinely hates to hear that term and sees it as a flag of, I probably should not listen to this person.
Well,
so
thanks so much. Thanks so much for that comment. And I'm always trying to learn more from individuals on the spectrum about how we can work together in these studies, because it's important to get that feedback. So thank you so much.
Yeah.
Okay. Is there any other question, or I can ask more questions? Okay, so my next question is that, so when you were looking at the flexibility across age, I really like these type of studies. My question is, have you looked at how much we have, like different, we know like different region, the cognitive flexibility, we can see different region and leave, say, integrate and segregate. I was wondering if there are any differences across different ages, either about the region that are part of this flexibility and the rate of it or not.
Yeah, that's a good question. I mean, there have been quite a few studies that are trying to answer this. And what we found was the difficult thing is that you have to get a task that adults find challenging, but that kids can still do. Because if the task is too hard, the kid says, they just can't perform the task at all. If the task is too easy, then the task doesn't make the adult have to be flexible. The adult can just automatically do it. They don't have to actually work very hard. So to find one task that can compare kids and adults at the same time has been the challenge for the field. So one of the thing I showed you, like the flexible item selection task, was is nice because you can use it in different age groups. But what we found is that, for example, the children, the adults can do it in 8 seconds, but the children, it takes them 12 seconds. It's just, they're slower. They're not up to speed. So it's very hard to find one thing that challenges both adults and kids at the same level. But at the same time, we think like the same sort of general systems are involved, frontoparietal, insula, ACC. It's just a matter of, I think, the dynamics actually between the regions that changes over development. And that's what we're trying to compare right now. So yeah, it's a good question. Wow, you have a great big group here.
Hi, everyone.
Hi.
Hi, Enrico.
Hi, everyone. I have a general comment, and also thank you very much for your presentation. Really amazing from my point of view. I'm very interested in the study of insula in the group of disease like frontotemporal dementia, in particular the genetic group. And I find very interesting your opinion on the flexibility concept, because in frontotemporal dementia, in particular in the presymptomatic stage, is very challenging to find out tests or cognitive evaluation that are sensible for the presymptomatic and slight alteration. So the concept of the flexibility could be very interesting in this condition. And on the other side, also the idea to study the dynamic and alteration of the subdivision of the instruments would be a very smart idea. So it would be. Thank you very much.
Oh yeah, thank you. Yeah, I was always inspired by Bill Seeley's work in frontotemporal dementia, and thinking about what are the effects of those regions really specifically degenerating in that disorder. So yeah, it's I think what's interesting about the insula and ACC as a system or network is that there's almost no disorder you can look at where those systems are not involved, right? So if you see it, there's so many meta-analyses now that are coming out that look at gray matter volume or connectivity or whatever, that you always implicate the insula and ACC. So there's something about those regions.
Yeah, there is a lot of meta-analytic data also on the involvement of the insula as a principal hub in psychiatric and neurological disorders. So it's a very shared alteration in this region.
Yeah.
Thanks again.
Thanks. Should I be looking at the chat here? Let's see. Oh yeah. So there's some comments. Thanks.
Yeah, can I ask another if I may, Army?
Sure. I have more questions myself too.
Well, then maybe you should take one. Good Lord, why are you indulging me? Anyway, just a curiosity, I am a big fan of your work. I've read and cited a lot of your papers, but naturally, since you're here, all of this information is leaving my brain. When you're developing populations, is it difficult to go from, say, kids that are very young, like 6 to 12, up into adolescents, up into adults, when you're dealing with brain normalization? Because I've seen a lot of work just coming out of UC Davis saying that the gyrification in the insula in particular changes quite a bit as a function of age. Has that been an issue or come up at any point?
Yeah, this is a great question. I may not have mentioned that our studies are a little bit older. I mean, the youngest we ever really get is 7 or 8. So we're doing, the sort of 8 to 12 age range. So it's an important question because the younger you go, of course, the less able the children are to complete a complicated task. So we have this sort of cutoff of really 8-year-olds can do the task, but younger kids wouldn't understand it or, wouldn't be able to really follow along. So we haven't gone younger than 8. And because of that, we've just been using normalization to the adult brains, based on the work by, Jay Geet and other about like the gray matter volumes and stuff kind of, reaching a certain stage at age 10. But yeah, this is a really important question. If you do go any younger, these sort of age-related structural changes, which our lab doesn't focus, certainly impact the network capabilities. I know there's groups who have as young as infant data, and following through the younger age ranges. So even though we haven't considered it in our own work, I think it'll be really important in, going forward.
Yeah, I think Norgal from UC Davis looks a lot at gyrification in the insula in particular, and her name comes to mind, as does I think her name is Schumann, but that's a lot of people.
Yeah, so the people doing structural MRI have the advantage that they can get sleep data, or they can get, really young kids to give pretty good data. Whereas, as you know, if you're getting functional data, the kid has to stay still, and we don't want to sedate them. So it's much harder to actually get some usable scans there. So we haven't caught up yet with structural folks.
Yeah, we had a little girl throw up in the scanner once. That was not a pleasant experience. So I feel your pain.
Yeah.
I have one comment, if I may, which is your point about the diagram where Tom Insel refers to your insula, which I think is a great strategy. But one of the things we've been looking at a lot is looking for homogeneous subsets within data, and then looking across cognitive or symptomatic, et cetera, information. And it's really an informative way to go. And there's all sorts of interesting sort of subclusters within the data. And it's kind of this chicken and egg problem, right? Where you're trying to figure out what we're studying, and we're also studying it at the same time. And so it's a little bit, circular, but I think that's where we are. I think it's really important to look at heterogeneity in general in all of these techniques. So just kind of a comment there.
Yeah, I totally agree. And in fact, we've been trying so hard to do things like connectivity-based subgroups in these heterogeneous populations. I have to say, with little success. Like we haven't found stable subgroups. We haven't found, some other research teams have definitely had more success with this. But when we've tried to say, like, parse individual connectomes and then say, well, here's one subgroup within autism, here's another one, here's another, we haven't come up with anything stable as of yet.
Yeah.
So one probably last question, we can continue our conversation in Slack. So Dolly is asking, are you interested in phenomena like mind wandering in relation with the state transition? How cognitive flexibility looks like in those phenomena?
Yeah, that's a good question. We have a paper out that was in collaboration with the Mishi Jaz group. I think it's in Human Brain Mapping 2019. And it's looking at mind wandering during a very easy task and how that relates to dynamics of the three networks I was talking about. So I really don't remember what we found, but we had one study where we did start to look at that. And I think it's a very interesting phenomenon to try to relate with these metrics.
Thank you. So I think we can continue our conversation and discussion. I have so many questions myself on the Slack. I really would like to thank you again for a wonderful talk, and thank you everyone for attending. So we'll see you on the Slack.
Thanks so much for letting your mind wander over to us.
Yes, it was my pleasure. I'm so glad to have this opportunity. And please do feel free to chat on Slack, and also just email me. I'm always on email. So happy to be in touch.
Thanks, and thanks everyone for joining. So
thank you.
Stay in touch.
Thanks.
Keep moving.
Take care. Bye.
All right.
Thank you very much. Bye-bye.
Bye. Thank you.